right, welcome back, folks, to part 3 of our notes on the nervous system. Today we're going to take a look at objective 3, or part 3, on nerve impulses, how we actually send a nerve impulse through the nervous system. Okay, first thing I want to take a look at, guys, is neurophysiology, and to note that neurons are highly irritable. So what does it mean if something's irritable? Well, if your little brother and sister came into your room right now and started making lots of noise and poking at you, would you kind of like get irritated with them and brush them away? You probably would. That would be irritability, right? That response to a stimulus. And that's what nervous tissue is. It's irritable. It will respond to a stimuli. The way it does this, one thing I want to mention, we've already talked about action potentials a little bit, but I want to do a little bit more right now. Action potentials, or nerve impulses, are the electrical impulses carried along the length of axons, and they are always the same regardless of the stimulus. Okay, doesn't matter if you got it's either full on or full off, nothing in between. Graded potentials, remember, they can be anywhere from down here, and they can ratchet up to all sorts of different levels between they're barely sending any signal, sending everything they can. That's it. Action potentials are either here or here. They're nothing or everything. And that is critical in how a nerve signal gets sent and whether it gets sent. And it plays a big part in how our brains are able to make decisions as well. And it's the underlying functional feature of the nervous system. Okay. How does a nerve impulse travel? Okay. Some input causes the dendrite to send a graded potential. How that's generated depends on the nerve. Is it a pain receptor? Is it a rod or a cone in your eye? Is it sensing the amount of protein in your stomach? Whatever it is. But something's coming in on that receptor, or maybe it's just getting signaled by another nerve at a synapse, right? The signal can be coming from another nerve as well. Okay, so let me draw this. Okay, so we got a dendrite coming in here. I'll just draw a few dendrites. These are going to be terrible dendrites, but they're long, thin, tapering, and they come down to the soma, or the cell body, right? Right here, and then we get to the axon hillock there. Okay, so the signal's coming down as a graded potential down the dendrite, around the cell body, and to the axon hillock. This is where the decision making, so to speak, in the nervous system happens. Okay, and it's reaching it, and this is all sending down as a graded potential. So this is a graded potential here. It's a graded potential here. It's just movement of ions, sodium potassium ions, across the membrane, like we talked about in the muscular system. But when it gets here to the axon hillock, we've got some voltage regulated sodium channels. Okay, and we've got all this sodium outside the cell. All these ions, I'm just going to do them little dots and things like that. All right, and that channel will only open up if there's enough signal. So if your graded potential is too small, that it will stay shut. If it's big enough, it'll open up. And if it's big enough signal coming in on the graded potential to open up, then the sodium potassium starts zipping through and generating a voltage. Okay. And if we can get those first ones to trigger, that is the decision-making part, right? If the signal's too weak, it doesn't open. If it's strong enough, it opens. If it opens, let's draw out the rest of our neuron here. And then I'll just draw one terminus down in the end here. There's our neuron. And this neuron's got sodium and potassium inside and outside, all the way down, big concentration gradient. Okay. Move to the next slide here. We have those on the outside, and what happens if we can get this first one to trigger, it is always going to generate enough voltage then to cause the next one to open up. And then sodium rushes in. And that's a charged particle. Remember from our last unit, movement of charged particles equals electricity. If that opens up, it's always going to open enough to let the next one open, the next one open, and so on, all the way down the axon until we get to the axon terminal. So back to the slide here. When the action potential is generated, it causes more action potentials down the length of the axon. Okay, all the way down here, like we're showing, sends a signal down the axon. Okay, when it reaches the synapse down here, okay, it's going to cause a neurotransmitter to get released, just like we saw with acetylcholine getting released, and that neurotransmitter can then zip across to whatever's next, whether that be a muscle or a nerve. I'll draw another nerve. We'll draw a dendrite here. Okay, and that neurotransmitter is going to get released, and it's going to diffuse across the synaptic cleft to some receptors on that dendrite, causing that dendrite to then send a greater potential. Okay, so that's going to happen down here. We got the same thing we saw in the muscles. We got little synaptic vesicles full of the neurotransmitter, be it dopamine, acetylcholine, what have you. Okay. Now, there's some things that can change the speed of this transmission. All right. If the dendrite has a larger diameter, a larger volume, we can actually get more surface area for sodium and potassium to zip through, generating larger currents and transmitting that signal faster. Kind of like a larger pipe will transmit more water than a smaller pipe. The other thing we can do is we can do, I'll do yellow for the myelin sheath here. If we put in a myelin sheath, okay, the Schwann cells, that's the peripheral nerve, blocking off that section, blocking off this section, blocking off this section. What's going to happen now is that signal is coming down, the sodium rushed through, and that signal generates a voltage, but there's no next one to open up here. So you got to go all the way down to this next one, okay, to open up, because this is blocking those gates. That Schwann cell is blocking that gate. So you go down to the node of Ranvier, right here, here's another node, and so the signal is going to jump between nodes. Okay, so it's kind of like if I was taking, trying to get out of this room, right? And I was walking along, I could take a whole bunch of little baby steps, baby step, baby step, baby step, baby step, to get to the end of the room. If I can't skip anything, I've got to do that. If I put in those nodes of Ranvier in the myelin sheath, I got to take big steps. And so it takes a lot fewer steps to get out of the room. I can get out of the room a lot faster. And that's basically a one way to think about how this nerve signal gets transmitted down this axon. Now, do you need a myelin sheath to do that? No. We got all that gray matter in the brain, right? No myelin sheath, but they're very short, so it doesn't take very long for those little baby steps to get to the end of the axon. If you got a long one, a long peripheral nerve like the sciatic nerve from your spinal cord all the way down to the bottom of your leg, many feet long, you definitely want to skip it along so you can get that signal down there quicker. So that's how a myelin sheath helps to speed up the transmission of a nerve signal. Now, I have some videos here that probably do a little better visual representation of what I just tried to draw on the board for you. I've got the YouTube links in the notes, so you can watch those on your own. I'm going to put them into this video right now, just so everybody gets one watch through, because if we were in class, I would definitely watch these and talk about them. So I'll put them in there. I may pause them at various times and talk about a couple of things that you're seeing. But the links are there if you'd like to see them. So let's take a look at the first video.
The fundamental process of neural transmission that underlies this action occurs in all neurons of the body. Neurons transmit this information through changes in the electrical potential of the membrane by the movement of ions across the membrane. An electrochemical gradient governs the movement of these ions, resulting in an electrical impulse. The resting membrane potential in a neuron, when the cell is not firing an impulse, is established by the unequal distribution of sodium ions outside of the cell and potassium ions inside the cell, making the outside of the cell more positively charged compared to the inside. The electrochemical gradient is established and maintained by an enzyme called sodium-potassium ATPase. When a neuron is stimulated, sodium ion channels open and sodium ions flow into the cell. This leads to a change in the electrical potential across the membrane called depolarization. The depolarizing electrical potential travels down the dendrites and over the cell body. Multiple electrical potentials will combine at the axon hillock in a process called summation. If the depolarization is large enough, an action potential is triggered. Action potentials are all or none electrical impulses that maintain their amplitude and strength down the length of the axon. The action potential travels down the axon when the depolarization of an area of membrane causes adjacent voltage-gated sodium ion channels to open. The influx of sodium ions results in membrane depolarization along the membrane. After a short delay, potassium ion channels open and potassium ions flow out, repolarizing the membrane. For the neuron to fire again, the resting membrane potential needs to be re-established. Sodium-potassium ATPase is used to move sodium and potassium ions against their concentration gradients, re-establishing the resting membrane potential. As the action potential moves down the axon, ions are diffusing only a short distance, allowing the signal to move quickly. At the axon terminal, the electrical impulse passes to another cell at a cellular connection called a synapse. The space between the presynaptic neuron and a postsynaptic cell is called the synaptic cleft. The presynaptic neuron contains signal molecules called neurotransmitters that are packaged inside vesicles. When an action potential reaches the end of a neuron, neurotransmitters are released by exocytosis from the neuron into the synaptic cleft. Neurotransmitters bind to the adjacent cell at receptor sites attached to ion channels. The channels open, allowing the movement of ions into or out of the effector cell, which alters its membrane potential, thereby transmitting the signal from the neuron to the effector cell.
Now the second video shows basically the same thing, but a little different take on it, so let's take a look at that.
A nerve impulse is an electrical current that travels along dendrites or axons due to ions moving through voltage-gated channels in the neuron's plasma membrane. Voltage-gated channels open and close in response to an electrical voltage, so they are affected by changes in electrical charge around them. When a neuron is at rest, a charge difference is maintained between the inside and outside of the cell. This charge difference is produced and maintained largely by active transport using sodium-potassium pumps. The pumps send sodium ions out of the cell and bring potassium ions in. While other channels allow some flow of potassium ions back out of the cell, the sodium ions cannot easily get back in to replace the lost positive charges. The overall result is that the exterior of the cell has a net positive charge and the interior has a net negative charge. The difference in charge between the interior and exterior of the cell is called the resting membrane potential. A nerve impulse begins when a stimulus disturbs the plasma membrane on a dendrite, causing sodium channels to open. Sodium ions flow into the cell, lessening the charge difference at that location. If the change is enough, it will cause nearby voltage-gated sodium channels to open. This allows so many sodium ions to flood into the cell at that location that the membrane there is depolarized, with the local region inside the cell having a net positive charge and the outside of the cell having a net negative charge. This affects neighboring voltage-gated sodium channels, which then open, moving the depolarization along the membrane. This moving depolarization is called an action potential. Changes occur behind the action potential to restore the resting membrane potential. The voltage-gated sodium channels close and voltage-gated potassium channels open. This allows a rapid flow of potassium ions out of the cell, repolarizing the membrane so that the inside is again negative and the outside positive. This is followed by use of sodium-potassium pumps to fully restore the resting membrane potential and to reestablish proper concentrations of sodium and potassium ions inside and outside the cell.
All right, now that you've watched those videos and thought about this and taken a look at the notes that I've got down, now I want you to do something similar to what we did in the muscular system, where we talked about how the signal goes from the nerve all the way to the muscle, and you had to document that, write an essay or a list of the steps, what causes what. I'd like you to do the same thing here now for nerve signal transmission. So from the time the dendrite sends a signal down, a graded potential down the dendrite and across the soma to the axon hillock, all the way down the axon with the myelinated sheath to the axon terminal, where it then sends out a neurotransmitter to the muscle, the gland, or the next nerve. I would like you to take some time and put together something similar, an essay, an outline, however you want to word that, for this segment. This will be the essay on our test at the end of the unit. That's it, folks. See you next time.